Nils, so do you see any positive effects of the BMPs that are released from beta cells on other tissues? For example, brown fat is known to be activated by certain BMPs. Do you notice if there's any good consequence of?
Well, I mean, we get asked this question a lot. Why do you have a factor that stops proliferation and inhibits differentiation, basically? And why this doesn't make any sense? But what most people argue with these things is it's actually a protective effect for the beta cell. Because the reason why beta cells die under these metabolic stresses is because they are beta cells. The alpha cell, for example, is not affected. It doesn't die. So by the fact that this beta cell has the machinery for recognizing glucose and responding appropriately, actually makes it susceptible to these, especially reactive oxygen species. So by downregulating these genes, you're actually protecting the life of the beta cell. Of course, you go into a state where it's not really functional, but at least it's viewed as a protective mechanism by doing this. So maybe the solution is not to relieve this, because then maybe you will just maintain beta cell function, and then they would be more susceptible to these effects. But it is a good question.
Nils, I have a question regarding pregnancy, because during pregnancy we have a transient insulin resistance. Why have you used the rat model, for example, to understand what's occurring during the course of pregnancy? Why is transition? There is some pregnancy-induced diabetes which is reversible. No, it's a very strange situation.
Yes, I mean, obviously, so there's two sides to this. The insulin resistance, I think, is quite well described during pregnancy. And the appropriate response there, both in animal models as well as in humans, is actually to upregulate the beta cell mass. So the few human data that does exist on autopsies from pregnancies show that the islet volume is about doubled in human pregnancy. Yeah. And it's also known, at least from the animal models, that this is a very quickly reversible. So shortly after delivery, these cells, probably through apoptosis, it's not really well described, simply resolve and disappear, and you're back to this normal state of, And of course, during gestational diabetes, again, this compensatory mechanism is inappropriate. It doesn't work correctly. But of course, there's very little human data on that. It's mostly from animal studies.
What's the source of that insulin resistance for gestational insulin resistance?
Felice is probably the better one to answer this question, but it has been studied, at least in animals to some extent.
The trophoblast hormones like HPL, which is prolactin, HPL, they modulate this is what we teach at school, that placenta secretes hormones like HPL, placenta lactogen, which modify the insulin resistance. But we don't know. Regarding inflammation, there is an inflammatory pathway which is being introduced recently.
But is the beta cell resistant as well?
That's not really known.
We don't know. We know that it's very reversible. You have gestational diabetes for 9 months, and then 1 week after, immediately your glycemia works much better.
And is that type of insulin resistance responsive to insulin sensitizers like TZDs?
Oh, yeah.
Yeah.
But I think
so it must be then involving the adipose depot in the end, because that's where a major target for TZD action.
No.
But I think, very few studies have been done in human pregnancy with these types of compounds.
But my real question is, there's been a couple papers from the Melton Group and Kiefer Labs on making in vitro beta cells, which, though not highly functional in vitro, become functional, according to their studies, when they implant them into the kidney capsule. Or is that a way that you see is a potential to try to rescue diabetes, or do you have a feeling about those studies?
I mean, those studies are obviously aimed primarily for type 1 diabetes, where there's an almost complete lack of beta cells and a complete lack of insulin production. So that's sort of the first stage for these types of transplantations with differentiated stem cells to different degrees. I mean, they are in clinical trials. I don't know how many, I think 6 or 8 type 1 diabetics have been transplanted with these types of differentiated or semi-differentiated stem cells, and we are just waiting for the results. But for the in vitro differentiation, more and more now, I think they're getting closer and closer to getting to something that looks very much like a real true primary beta cells in vitro. And interestingly, the last step of their maturation is actually BMP inhibitors that they actually add to their cultures to get the full or as full a maturation as they can get in vitro. It's actually through inhibition of BMP. So,
Bill,
so maybe there's a connection.
Very quick one.
Another way to ask Ron's question in sort of a contemporary genetic way would be, what do the GWAS show in gestational diabetes?
They show very much the same variants as you would see under normal, in normal type 2 diabetes. So the variants are very much the same. And I think it's simply a matter of pregnancy being a very accelerated form of obesity that you just develop during 9 months instead of, for most of us, 10 or 15 years.
Well, another way to look at it is a stress test for the pancreas. I mean, pregnancy is an excellent stress test for the pancreas. And because there is a prediction for later type 2 diabetes. Classically.
Absolutely.
I'm not sure that the GWAS show exactly the same as type 2 diabetes. I have a recollection that there are some different genes, and we ask these in terms of this modifier, that pathway of knowns. The right way to ask the question, I think, now that we have 185,000 patients available, is what are the loci that determine this? Because that's how you know find out the known, unknown, you find the unknowns in this pathway. A related question, though, relates to the therapeutics and the binding proteins. What's the role of follistatin here? Because if you knock out FSTL3, you get enormous beta cell hypertrophy, you get hypoglycemia, and you get huge upregulation of gluconeogenesis, implying that there are some of these proteins where follistatin plays a role by blocking. Do you have a profile of this? And it's FSTL3, the one that stays intracellular, not the secreted form. Do you have any idea of how that maps on the differential BMP pathways that you're looking at in terms of administering follistatin or anything as a sort of an endogenous antagonist?
Yeah, we have not done the experiments with the follistatin or the FSTL3, but you were mentioning the intracellular part of it anyways. What we do know about activins, because they are the major binders in this case, is that they have very unfortunate species differences from mice to humans. So we see almost opposite effects of activin on beta cells from humans and mice. So therefore, we are a little bit concerned with the knockout studies, because when we try to replicate it, and others, you often see the exact opposite in the human islet. So there's really something we have to figure out there, what's going on. But I know there are individuals that are now trying to address this, where they transplant human islets into mice and then try to do the treatment and compare it to the endogenous beta cells in the mice.
Thanks very much indeed. We have to cut the discussion right now. Thank you very much.
Thank you, Nils.
Thank you very much.